Welcome back, everyone, for another episode of Max Planck Florida's Neurotransmissions podcast. I'm Joe Schumacher. I'm joined by my colleagues, Dr. Jeremy Chang. How's it going, Jeremy?
Good.
And Dr. Lesley Colgan. How's it going, Lesley?
Very good, Joe.
We've had another really great Sunposium meeting here in 2023. We just had 3 fantastic interviews with some of our guests here who've presented at the conference. So tell us a little bit, for people who don't know, a little bit about what Sunposium is, what the focus is.
Sure. Sunposium is a conference that we hold every 2 years. It's focused on neural circuits, and it's a really nice conference because it's sort of medium in size, so I think the scientists really get an opportunity to interact with some of the speakers. We have a really great lineup of speakers this year, and I think it's been so far a great conference.
Yeah, I think it's been fantastic. Shout out to the organizers for putting on such an amazing conference. Jeremy, you actually presented a poster last night. How does this Sunposium compare to some of the other ones you've been to in the past?
I think Symposium's kind of nice because it's a little more intimate, but also there is a lot of diversity in sort of the audience that you're presenting to and interacting with. So, you're talking to like undergraduates that are looking at where they're headed in neuroscience, and established PIs that have won lots of awards and or have really progressed the field forward. So I think it's an interesting mix of both interests and also backgrounds at the conference, and it's kind of nice that it's right in our backyard.
Yeah, definitely. I mean, it's a beautiful setting here in downtown West Palm Beach. So we have 3 really great interviews lined up to share with the listeners. So I guess we'll just move right into that, and we'll be back to chat after we talk about these interviews.
So we're here today with Dr. Denise Cai. Dr. Cai is an associate professor of neuroscience at the Friedman Brain Institute at Icahn School of Medicine at Mount Sinai, and she studies how our experiences are recorded as memories and how these memories change over time and with further experience. So thank you so much for joining us today, Dr. Cai.
Thank you so much, Lesley, for having me.
Before we really dive into your science, I just want to, start really basic.
Yeah.
And maybe you could tell us what exactly is an ensemble and how does it relate to memory in the brain?
Yeah, so a lot of studies have shown that there are, when we experience something, so this conversation we're having here, right? The scientific literature would suggest that we are forming a memory of this conversation, and that this memory is stored in a subpopulation of some of the cells in your brain distributed across different areas of your brain. And the idea is that that memory is stored there such that, tomorrow when you're like, oh my God, that great conversation I had with Denise, and you recall that memory, right? Many of those same neurons might be reactivated, and that neural ensemble or that group of neurons that's thought to be active when we're chatting today, and then when you're thinking back to a conversation tomorrow, it's thought to underlie that stable memory representation.
So experimentally, how do you actually get at identifying some of these neuronal ensembles? I know you have done a lot of really great work in technology development, especially making some of these technologies accessible and open source. Maybe you could sort of talk us through some of the technology developments that have allowed you to sort of dive into this research.
Yeah, so I'll answer your first question because there's like a couple questions embedded in that. So the first question is, how do we do this? How do we look into the brain? And so we and others have developed these tiny miniature microscopes, like the size of like the part of your pinky, right? And they're just, some plastics and lenses and optics that we put together in the lab that were like homegrown, DIY, do it yourself, right? And it's the camera sensors, what you find in your cell phones, what you and I carry around all the time. And these tiny miniature microscopes, about maybe 2.5 grams, we can put it onto, living animals. So things like I study mice, other people study other species. And then we use these fluorescent indicators. We genetically encode these cells. And when the cells are active, they flash green, right? So then we use a camera to capture the movie of the brain. And so we can peer into the brain while animals are freely behaving, interacting with the world, forming memories, recalling memories. And so it's with these tiny miniature microscopes that we're able to peer into the brain and look at their neural activity in real time. So your second question was asking about developing the tools. Is that what you want me to talk about next?
Sure.
Okay,
great.
Yeah, so when I was a postdoc at UCLA, I really wanted to study how is it that memories interact across time. And in a lot of animal studies, right, typically an animal has one really salient experience where like that's the memory. But our human experience every day, we have lots of things that happen all the time. And so I want to understand how is it that these different experiences get encoded in the brain and how do they interact with each other. And there weren't a lot of tools that allowed us to look into the brain while animals underwent lots of different experiences. And there was a tool, this miniature microscope was developed by this lab at Stanford. And we're like, whoa, mind blown, right? You can like take this huge microscope that's typically like the size of a desk and then super miniaturize it and put it on a mouse's brain while the animal does cool stuff. So we were like, wow, that'd be super cool. At the time, this technology was super expensive, like prohibitively expensive, hundreds of thousands of dollars. And, we thought, wouldn't it be awesome if we could just develop it in an open source way and make it super affordable to everyone, and we shared it. And we thought okay, maybe 10 labs would be interested. And if 10 labs were interested, then that'd be worth not only developing the system such that anyone can use this microscope system to image the brain, but we also developed a platform for sharing the system. So we built this wiki and anyone could access it. Either developers who were on the engineering side could further innovate on top and bring it back to our research community, or people that are more like psychologists, biologists that want to understand the brain, they want to plug and play, they could just download everything and use as is. And so we developed this platform. So we launched this in 2016. And since then, it's been a few years since then, now it's 2023, I think we have something almost 1,000 labs around the world have now used our open source miniature microscope to understand brain behavior relationships.
That's really exciting. What a way to sort of magnify your work in science, really expand.
Yeah, I think some of the really core philosophies that through this project that I developed, but also, that is really at the center of what my lab does, is increasing accessibility. And it's really this belief that, these hard questions that we have not answered for a long time, right? It really takes people from different backgrounds with different perspectives and us all coming together with our different ideas to forming a new idea. And so this is really kind of the belief, the core belief that we center our open source tool development on.
So you got interested in this miniscope for tracking these neural ensembles over time.
Yeah.
What aspects about these neural ensembles were you interested in?
Yeah. So I remember that we were really interested in how is it everyday kind of memories form and interact. And we had this intuition and some really, molecular and cellular data suggesting that if two memories happen close in time, that they would somehow be related. Because when we recall memories, if you recall one memory, you're much more likely to recall another memory that happened close in time versus separate in time. And this is just kind of how our episodic memories are formed over time. And so we wanted to understand what were the principles that allow memories to be related. And what we found was that when two experiences happen close in time, even though they're really different memories, and at the time everyone thought different memories go into different neural ensembles, and this is how the brain knows it's memory A and not memory B. Well, what we found was just by having these two memories close in time, they were more likely to share some of those same cells representing both memories. And this is how, at the biological level, that when you recall one memory through that shared neural ensemble triggers a recall of another memory that was encoded close in time.
So this is you smell apple pie and you think of your grandmother
Yeah.
or something like that, right?
Yeah.
And what is sort of the time window that this linking occurs?
Yeah.
I mean, if you have an experience, 2 days apart or a couple hours apart,
Yeah.
how does that affect it?
Yeah, so great question. So the scale is really quite invariant in terms of all the ways that we can link things across time, right? And so what we really focused on were how distinct experiences can be linked across many hours. And up until that time, we thought that across hours, different memories would be formed in different neural ensembles. More recently, we've been interested in another way to link memories. So what we showed before in 2016 was that memories encoded close in time were more likely to be linked, but memories that happen across days not linked. So then some recent studies from my lab suggest that, well, even after memories are formed, the brain is so dynamic and our memory systems are so dynamic that there are opportunities for memories to be updated such that they can be linked at a later time to other memories that may be relevant so we can make causal inference about the world.
Wow, really interesting. So one thing that I know that you have been particularly interested in is how these memory processes such as linking change actually as we age.
Yeah.
And, I think it's pretty well known that as we get older, unfortunately, our memories seem to have some deficits.
Yeah.
Are there specific deficits in memory and aging that you're interested in studying?
Yeah, so we're really interested in normal aging and particularly in middle age. And I think a philosophy of ours is that, by the time there's so much neurodegeneration or plaques and tangles with Alzheimer's, it might be too late. So can we track how memories change at an earlier time point? Are there markers, behavioral or biomarkers, that can predict future cognitive decline? So my lab is really interested in, how aging goes right with middle age. And so we typically are looking at this where these mice that we study, they can learn single things just fine, right? Learning one thing, no problem. Spatial maze, this context I got shocked in, they can learn these single experiences great. What is challenging in mice and in humans is being able to relate this and update that information flexibly with new information. And we find that that ability to update and change flexibly, dynamically, change in real time, that is really hard for the aging brain. And that these cells are not quite as flexible to update these neural ensembles that we've been talking about.
Could you explain a little about why that loss of flexibility is an issue with aging, or?
Well, I can say why it leads to behavioral challenges of flexibility, right? So if the cells are firing when animals are in context A, and then now they're in context B, and those same cells are firing, animals get confused and they generalize context A to context B. What is driving this? So we think that in hippocampus, so there are areas that that are hyperexcitable, even they're upstream of hippocampus, like interminal cortex, and aging that's shown to have increased excitability. Now, what I find super fascinating is that within the hippocampus, this region, there is this region called CA1, and I've documented, and others as well have documented that there's decreased excitability, meaning that these cells are less likely to fire. Now, what's really interesting is that there's this upstream region that feeds into CA1 called CA3. And this region has high excitability and it fires a lot. So then we started thinking, well, which one, there's a chicken and egg problem, right? Is it because there's so much firing from CA3 into CA1 that CA1's like, whoa, hold a minute, let's slow it down and let's decrease and dampen excitability so we don't go into seizure mode? Or is it that because, CA1 has dampened excitability that CA3 is like, what's up, CA1? And having to fire more, right, to be able for it to receive its input. So we're very interested in this chicken or egg problem. Is it CA1, CA3, is one leading to other? And again, going back to, can we prevent this from happening? We can prevent the decreased excitability in CA1 or increased excitability in CA3. Can we then alter the effects of the other? Yeah.
So regardless of whether it's sort of which comes first, it seems like this problem of generalization might be particularly behaviorally a problem in contexts where there's maybe a dangerous, a dangerous experience that someone might go through. So if they generalize that experience, that dangerous experience, beyond that single context and generalize it to other contexts, that seems like it could be problematic. So our memories that are, dangerous, for example, encoded in the brain in a different sort of way, and how is that related to these deficits that you're seeing in aging?
Yeah, so what we had documented was that in young mice, so as I mentioned earlier, they're able to link memories that happen close in time across hours, but distinguish memories that happen further time across days, right? So this temporal distinction is really important, and you don't, mislink things that are across time. And so what we found in our middle-aged mice is that while they can learn single experiences just fine, they did not link temporally, and so they did not distinguish if memories were experienced 5 hours apart or 7 days apart. But what was interesting is that these mice, as you had alluded to, they tend to generalize more their fear, right? So they were shocked in one context, in context A. They're more likely to generalize it to any circumstance. So, one interesting question is, what's the evolutionary reason, right? And it might be like, better safe than sorry. If you can't temporally specifically link your memories across time, if it's really bad, maybe it's just better to avoid both of those potential arenas. So, this might be kind of a compensatory mechanism that we see in aging, this overgeneralization. So the other pet hypothesis I have about generalization with aging, so this idea came when my son, at the time he's 3, he's 10 now, but he is 3, and we're playing this it's like Go Fish game, but it's with Nemo. And so it's a visual search game, and you have these cards, and you have to remember, like, here is Nemo looking left, and you have to pick up another card that's Nemo looking left. And then if it's not, you put them back, right? So it's a visual memory game. And I just remember thinking every freaking card is a Nemo. It's like Nemo looking left, Nemo looking half left, now Nemo looking straight. And to me, like, my brain is like, they're all just Nemos. And my son's like, no, this one is with mouth half open, and this one with mouth quarter open. I'm like, how is it that his brain is able to differentiate all these things that look similar to me? And I begin to wonder, is it because over time we have all these different experiences, right? And so we have all these different memories just stacked on top of all of our synapses are used. And so there's no more, quote unquote, real estate for, new ideas that are not interacting with other ideas. And so I was like, I don't think my son's that much smarter than me. Maybe it's just because he has like a blank slate for all these different memories, and so they don't get confused with each other. And I wonder if in part with aging, it's just because we have so many memories in our brain, they start to stack on top of each other, and maybe this is why we get confused or generalized.
Well, we're about out of time,
Okay.
But before we finish, I do want to ask sort of what's next? What's your next focus? Is there anything new and exciting in the lab that you're, can share a little bit with us?
Sure. So the question that keeps me up at night, right, is similar to the things that we've been talking about today. But the overarching question is, how is it that we accumulate what seems like an infinite number of memories across our lifetime in our finite number of synapses and cells in our brain? And so I think really my lab is developing both the tools in terms of hardware development and the analysis tools, as well as the behavioral paradigms to really look in more naturalistic settings. And so now we could record so many thousands of cells simultaneously in the brain as animals are experiencing lots of different things, and we can record their brain state, whether they're sleeping, they're awake, across their entire lifetime. Could we get a better understanding of how our brain is able to maximize storage capacity of memories across the lifetime? So hopefully in 5 to 10 years, we'll have a better answer.
Fascinating. Thank you so much.
Here at Sunposium 2023 once again, and our guest right now is Dr. Ugur Dag. He's a postdoctoral associate at the Picower Institute at MIT in the lab of Steve Flavell. Ugur, thank you so much for joining us on the podcast.
Yeah, thank you for having me. It's a great pleasure.
And it's really nice to meet you. We've never met before, and I was introduced to your work being on the selection committee for the Sunposium abstracts, and your work sounded really exciting, and it was really exciting to hear you talk today. So tell us a little bit about your work. You're studying the functional organization of the serotonergic system in C. elegans. What is it about C. elegans as a model organism that's interesting to us in general as neuroscientists, before we talk about serotonin specifically?
Yeah, I think one of the most powerful thing about the C. elegans that we have a full connectome identified already. So there are 302 neurons, and we all know the interactions between these neurons synaptically. So this is very nice and cool. At the same time, we also have a very powerful genetic tools that we can manipulate the activity of the neurons. We can either activate them or inhibit them, and then we can just check the behavior correlates. And maybe another nice thing about them, they are transparent. So when we wanted to really identify and see some imaging and see how neurons are being activated, it also makes us life easier to monitor that kind of experiments too. I think in combination of these genetic tools, full connectome, and also the ability that we manipulate them, it makes them a good system to study neuroscientific questions.
Yeah, and you're really interested in neuromodulation and
Yes.
how that might affect behavior. And one thing that I'll admit I didn't know, there are actually only 3 serotonin neurons
Yep.
in the C. elegans, and each have sort of a unique function. And I just, thought it was incredibly amazing to think about, a single neuron releasing serotonin and having such a dramatic impact on behavior. And perhaps one of the ways of this, which you mentioned in your talk today, is that it actually releases serotonin through volumetric release as opposed to more synaptic release. So maybe you could explain basically what volumetric release is and sort of what implications that has.
Yeah, I think that would help the release serotonin from NSM have a effect on the more distant neurons and for longer time scales. For with volumetric transmission, we can achieve that. And NSM has a unique feature, and then it's located in the pharynx of the animal, close to the head. And then where it extends its neurons, it's very close to the neuropils. Extensions from other neurons, and they are very dense. They are really, coming from multiple neurons, and they are in the one core area. And then once serotonin released from NSM, it may act on these many neurons through the nervous system, and it can have an effect more a longer time scale. I think that one of the key information about that we know about NSM and serotonin release from NSM.
So you mentioned NSM. NSM is one of these specific serotonergic neurons. The way you're describing it, it sounds like NSM has this big area over which it's releasing serotonin, and then all these other neurons have little receptors in that area, so it can sort of target a lot of things at once. In principle, if we want to understand how, say, serotonin modulates behavior in C. elegans, if you were to say just optogenetically stimulate or stimulate in whatever way NSM, what would that do to the animal? If it just sort of dumps a bunch of serotonin into this area, what happens?
Yes. So actually, even without any optogenic manipulations, we were able to see when the animals are finding a good food source. Actually, by the way, they eat bacteria.
Okay.
And then when they eat it, some particles from the bacteria, it just binds the specific receptors of this neuron, NSM. And then that leads to the activation of the neuron. And even before any manipulations, we were able to know that. But what key thing that we didn't know and we wanted to study, it's just being activated, and upon this activation, it may release serotonin, and then animals will start some behavior changes. Especially they stop moving, they have a low locomotion speed, and they start eating even more, increased feeding rate.
Okay, so you actually can see naturalistic behavior in the worm. It's going around, it's picking up bacteria. Yeah. The bacteria itself is causing the release of serotonin, and then the animal slows down. Yes. So is the slowing down because it needs to eat? Is it basically it's just here's a snack. I'm going to stop moving around. I'm not going to forage anymore. I've got my food.
Yeah, exactly. This is the point. Yeah, okay. Because when there's no food, the animals just move very fast, probably just trying to find a good food source. But once they find it and sample it, perhaps they just can decide, okay, this is a good place, so I should slow down, I should dwell on the food, I just eat enough, and then once I'm not hungry anymore, I can start moving again. So there's there's always a frequent transition between these two behaviors, and serotonin is regulating, especially from this roaming to the wearing transition.
That's really cool. It's so we just saw the Peter Groos Award-winning talk from Dr. Yartsev. He was talking about naturalistic behaviors and how, there's this idea that naturalistic behaviors necessarily must be noisy and complicated, but both his talk and yours demonstrate that very simple behaviors that are important for sustaining the life of the animals can be really easy to characterize, and then you can witness what the neural circuit is doing in real time. So that's I guess another shout out to C. elegans for being a really powerful tool.
Where the behaviors might be more simple, how that actually is occurring and the mechanism is actually, as we learned from your talk, very complex. And you actually mentioned that sort of, the serotonin is being released in this volumetric way to influence a lot of neurons. And unfortunately, these because it's complex, these neurons actually have a lot of different receptors that can interact with this serotonin. So maybe you could just give us a little overview of that.
Yeah, actually, that was all the goal of my project. I wanted to understand. Okay, we know serotonin is released from this neuron, but how it may act on many neurons with many receptors, with many different types of receptors. So I wanted to identify the laws of each of these receptors. As you say, I mean, there are many receptors, and then first we wanted to see if we can dissect their laws in the behavior. Do they contribute to the behavior? Or do all of them do that? Or maybe some of them are more important than the others? So we wanted to start with this. And we created mutants. So far we know there are 6 receptors exist in the nervous system. Maybe there are more, but I mean...
6 serotonin receptors.
6 serotonin receptors. And then we just wanted to knock them out and see how behavior is impacted. And we did this in a way that we can delete them, all possible combinations. So we had single mutants where we knock out only one receptor, but then we had double mutants where we took 2 of them, triple, quintuple, it goes like that. And then we actually found that they have a different laws. So for example, some of them are really important, and when they are intact by themselves, that is, if they are expressed only one type of receptor, let's say MOD1 is one of them. If the animal express only this receptor, they were able to still slow down, even though there are no other receptors exist. But there were other cases, for example, another receptor, there is only one type of receptor, but animals cannot slow down. So basically, they have a different driving forces, different weight in the slowing. And more interestingly, actually, there is also some kind of interactions between them. It's not linear. So for example, if I have double mutant, they will be more severe than single mutant. It's not the case all the time. So there is some weird genetic interactions between them. They can be inhibiting each other, they can be combinational effect, or they can be more synergetic effect. So it's a little bit more complex than we might think of. But we were able to have a model as well, if we can predict how animal can slow down based on which receptor they are expressing. And we have some information about that too. But in general, it's quite complicated, as you say.
And so when you're doing these types of selective combinatorial knockouts and that sort of thing, you also showed, I think in your talk, that each of the different receptors sort of acts over different time scales or has different time constants to it. How do you measure that? How do you sort of determine what times of the slowing down each receptor is responsible for?
So what we did actually, we activated the neuron NSM that we have been talking about with optogenetic tools. So we were able to just activate this neuron, but nothing else. And then we just measured the behavior changes based on this activation.
In the knockouts.
Yeah, exactly, in the knockouts. So, and then we just saw that the animals which are expressing only one receptor, let's say MOD1, they were able to slow down upon the NSM activation. And then they maintained that state. When we activated longer, the animals slowed down longer. They just follow the activation paradigm. But there were some other receptors, for example, they were able to slow down, but they cannot maintain that state. They go back to their baseline speed immediately. They cannot sustain this slow state, and even though NSM is still being active. So that kind of suggests us that these receptors might be sensing something different, and then might be regulating different phases of the behavior. Because slowing basically, the animals just move at a certain speed, and then they should slow down to some speed, some lower speed, locomotion speed, and then they maintain that state, and then they go back to their baseline speed again. So there are some different temporal phases, and it seems like these receptors are regulating these different time phases.
So the release of the same neurotransmitter is having really diverse effects on this one behavior. And there's, I think, another layer of complexity even on top of this, if I'm correct, is that not only do you have all these different receptor types, but they're expressed in different neurons, right? So trying to dissect the effects of these behaviors could depend on either the receptor types that it's interacting with or the specific neurons that it's actually activating. And you actually, I think, did a really sort of interesting and cute experiment to try to address this.
Yeah, you're right. So, for example, when we were comparing these two types of receptors, we were wondering what's the difference? Especially when I talk about these two receptors, they are both inhibitory. So and then they should be doing something similar to each other in terms of this. But why are they different? So we ask the same question as you raised. maybe the molecular properties of these receptors are important, or maybe the cells that these receptors are existing is more important. And we wanted to test this. And we simply just swapped their expression. We just express one of the receptors in the cells which are originally expressing the other receptor. And then we did the same experiment. And then we saw that rather than molecular properties, actually the cells which express these receptors are more important. And then it was kind of interesting for us. It may raise the question, why is that? And just we think that maybe the location of these receptors in the network, it might be different. Some of them could be in the locomotion circuit, enhanced in the locomotion circuit, or some of them should be enhanced in the feeding circuit. So there is also another complexity that where in the connectome they are being expressed seems to be, remaining question by the time.
And then the final layer is, because this is a small animal and it's clear and you have all the cells and you have pan-neuronal labeling of things, you can then look at how all of the neurons are responding during serotonin release and that sort of thing. What are some of the interesting things that you've seen when you're able to do like whole animal imaging, basically, all neurons at once?
Yeah, that was we were kind of interested in that, as you say, and then we specifically wondered how serotonin released from NSM neurons can have an impact on the whole brain. And we wanted to test this in a more naturalistic way. What we did, we developed a system in the lab where we can monitor the animals' behavior. We let them move towards the food source, and once they find the food source, there will be some behavior changes, and we were able to capture these behavior changes. And at the same time, we express pan-neuronal GCAM, so we are able to detect the neuronal activity in every neuron, and then we can correlate this with behavior correlates. So and then that gave us a clue that also is more importantly, when animals are on the food, we know that NSM will be activated because they will be eating, the NSM will be activated. So if we just compare the activity of NSM with the rest of the brain, then we can see some causality or maybe some correlation between the NSM activity, thus serotonin, and the rest of the brain. So that gave us information about whole brain dynamics, how they are changing upon food encounter.
This is really great, and it's just amazing to think, we try to take one of the most simple systems, and it just reminds you of the incredible complexity of the nervous system, and but how, even a small number of neurons, in this case one neuron, can have such a dramatic effect through so many different mechanisms and really affect behavior.
It's almost like there are no simple systems.
Yes, exactly.
I mean, systems are systems. Some systems are complex. There's always something. I guess before we wrap up here, one other question I have is just sort of like on the nature of serotonin in general. So this is a molecule that's, important for nervous systems from worms to humans. And it seems like, when I think colloquially about what does serotonin do, it's important for like how I feel. I take this pill, it affects my serotonin, and I feel better, this sort of thing. So in the worm, it seems to be regulating something down to the level of like feeding behavior. So what does that say about sort of the evolution of how nervous systems have like incorporated this molecule? Like it seems like it has such a diverse functionality across so many different taxa. Do you have any thoughts on why it might be doing one thing in one system and why it might be doing something completely different? Are these just bootstrapped processes that sort of get elaborated the more complex or more cells we have in our brain? Or
actually there are some similarities between species as well. So when I say regulating feeding in worms, for example, and then there were some examples that showing serotonin may be also regulating feeding behavior in mammals as well. But I think more globally, if you think more globally, serotonin is important for changing the state of the animal, right? As I described in the first part of the talk.
Yeah.
So it's just important for transition from one behavior, like high locomotion movement, to the low locomotion movement. So, and then it may just regulate this transition. And it's shown that actually it also regulates that kind of transitions in the mammals too. For example, it regulates sleep-wake regulation, or eating regulation, hunger, or thirst. So it's involved in that kind of behavior changes. Maybe there are different paradigms.
I see.
But in general, actually, they are doing the similar things in high organisms too. I think the lore of serotonin in terms of this, also conserved among the.
Yeah, it's sort of like the commonality is changing in state, and how that affects different organisms might be similar or different. How do you look at it? That's very interesting. Yeah. Well, thank you so much for sitting down to chat with us about your work. I think it's really great that you're down here at this conference, and we get to meet new friends and see cool new science. So I should mention your work is now up on bioRxiv. It's in revision, right?
Yes, it's in the revision. So actually, we submitted our revised version. So peer-reviewed version also can be online soon.
Awesome. That's very cool. Yeah. Very cool. Well, Dr. Da, thank you so much for sitting down and talking with us.
Thank you very much for having me.
Have a great rest of your conference. Sitting down with a really exciting guest. We're really glad that he decided to talk to us on the podcast. Dr. Sergiu Pasca is a professor of psychiatry and behavioral sciences at Stanford University. He's also the director of the Stanford Brain Organogenesis Program. Dr. Pasca, thank you so much for sitting down and chatting with us.
Thank you so much for having me.
So, first of all, welcome to the show. You're really interested in understanding human brain development, and that's an extreme challenge in neuroscience. We've interviewed a lot of people on this podcast. Everybody kind of has the starting point of being fascinated with the mind and our own brain and how it comes together in development. But everybody has generally different approaches, and one of the most conventional ways is to use genetic tools like flies and C. elegans and mice to, understand circuits in the brain in general, but you take a very different approach. You're very much focused on what human tissue is doing. So can you tell a little bit about how you study human brain development without going to the crazy extreme of studying human brain development?
I'm a physician by training, and my interest has been primarily in trying to understand psychiatric disorders, especially those that impact neurodevelopment, such as autism and schizophrenia. But the main challenge that we've been having is that we cannot access tissue from patients. And many of the genes that are associated with these conditions impact brain development or express very early in brain development. So this has been a challenge. How do we actually, in a causal way, tackle the role of these genes? And there's no doubt that animal models have been incredibly useful, and they will continue to be very useful. But there are unique aspects of human brain development that most likely are related to the psychiatric conditions, and to a large extent, many of the psychiatric conditions are uniquely human. So it became clear over the last few years, and especially in the last decade, that we will need also human cellular models to complement many of the animal studies. And that's why we've been primarily focusing on, certainly not recapitulated in total, but in isolation, critical aspects of human brain development. And that's exactly what we've been doing for more than a decade now.
So part of what's intrinsic to your work is the development of these so-called brain organoids and assembloids. Can you talk a little bit about what goes into creating something like that and what they are exactly?
Well, all of this effort started really after Yamanaka discovered cellular reprogramming, now more than 15 years ago, when he showed that you can essentially take somatic cells, push them back in time, turn them into stem cells, and then of course you can take those stem cells and then guide them to become various cell types, including cells of the nervous system. So it became clear that it will be possible to make human neurons and human glial cells from patients in a dish. And that's what we did very early on, made some of the early neurons at the bottom of a dish, around 2009 or so from patients with autism. And that was certainly very good, and you could discover especially phenotypes that would be related to neurons themselves, like the morphology of a cell, the electrical properties of the cells. But the nervous system develops over a very, very long period of time. Right? It takes at least 140 days to make just all the neurons in the cerebral cortex, and that's without making all the glial cells. And if anything, brain development continues for decades after birth. And what was really challenging for us in the beginning was that we had these neurons at the bottom of a dish, and you could maybe keep them for 100 or so days, but then they would peel off and die. And so we thought, would it be possible to just not let them sit at the bottom of a dish, but rather keep them in three-dimensional culture so we don't have to plate and replate them. And that's essentially what we did. We just moved them to a place that were coated in such a way so the cells cannot stick to the surface, aggregated them so that they can make tiny spheres, which is why we initially called them spheroids, because they're beautiful spherical structures, clumps of cells. And then we very soon realized that not only we can keep them for very long periods of time, we can keep them for hundreds and hundreds of days, but they also self-organize. And they recapitulate development in a very stereotypical way. So they'll first make the key progenitors of the nervous system, then they'll start making neurons in a sequential manner, then they'll switch and they'll start making glial cells. And collectively, this three-dimensional cultures that are self-organizing became known as organoids, and that these advances have actually been happening across multiple branches of medicine, not just in neuroscience, in the last decade.
So just to make sure I understand, so you can actually recapitulate some of the cellular diversity as well within a single organoid that we see in the brain.
Right. But generally, organoids resemble specific domains of the nervous system. So if you actually generate the stem cells or the progenitors that will generate cell diversity in that specific brain region, then yes. Of course, the challenge with the nervous system it's not homogeneous. It's unlike the liver, almost any part is pretty much the same. In the nervous system, we have immense cell diversity and cells communicating across brain regions, which is why we came up with the approach known as assembloids, where you essentially make these different brain regions, and then at the right time, you put them together to either communicate through cell migration from one region to the other, or to form connections at a distance. And then that really summarizes the generation of neural organoids that resemble specific brain region, and of assembloids that look at cell-cell interactions across brain regions.
That's really exciting. And as you mentioned in the beginning, sort of your goal for a lot of this research is to be able to understand human developmental disorders. So can you maybe just walk us through one particular example of how you've used this approach to understand some disease phenotype?
Yeah, so we've been studying now more than a dozen conditions associated with disease, mostly genetic, some of them environmental as well. But one in particular that I can highlight is called Timothy syndrome, which is a very, very rare form of autism and epilepsy. We only know of about, maybe about 50 patients worldwide, so it's very, very rare. But it's a devastating condition. And what's interesting is that this condition that, again, has epilepsy, has autism, intellectual disability, is caused by a single point mutation in a calcium channel. There are calcium channels on neurons, as you probably know. They essentially sense changes in the electrical properties of a neuron. When neurons communicate with each other, they open and they let calcium in. And that's one way of turning electrical signal into chemical signal inside a cell. Now, in patients, this point mutation makes the channel stay open for just slightly longer. And by doing so, it lets more calcium get inside the cells. And that's essentially that's all we knew about this mutation. But of course, these channels are expressed in excitable cells. So in order to study this disease, you need excitable cells, meaning cardiomyocytes of the heart or neurons, which were very difficult to make before. And so when we made neurons from patients and we looked at calcium signals, now more than 12, 13 years ago, we've discovered indeed that calcium will go in the cells, but then it will stay for much, much longer. So it will take the cell much longer to get the calcium in. And that was the first defect that we've identified in this patient. But subsequently, as our models became more complex, we started discovering pathophysiological process that may be responsible, for instance, for epilepsy in this patient. So if you, for instance, perhaps or maybe if the audience doesn't, but the cerebral cortex, the outer layer of the brain, has both excitatory as well as inhibitory cells. And what is interesting is that inhibitory cells, which are the ones that put a brake on excitatory cells, are not actually born in the cerebral cortex, but they're actually born in very deep regions of the forebrain. And then they literally have to migrate for inches and inches over many, many months, and in humans up to the second year of life, to populate the cerebral cortex. And so what we've discovered is that if you make forebrain assembloids that have the cortex on one side and the ventral forebrain part that has all the interneurons, and you integrate them with each other, then these interneurons, these inhibitory cells, will start migrating towards the cortex. Now, what is interesting is that when you make forebrain assembloids from patients with Timothy syndrome, these interneurons, these GABAergic cells, will actually move much more often. So they will undergo these jumps much more often, but every single time they jump a shorter distance. So in the end, they're left behind. So that could cause essentially a delay in the arrival of these neurons in the cortex, and perhaps they won't put a brake on inhibition in the cortex at the right time.
So that's like kind of a perfect example of like a small change that has a drastic effect at very specific, important developmental periods that you can sort of visualize using these
Absolutely,
assembloids.
yeah.
That's really fascinating.
It's interesting because this process of migration happens in the second and third trimester. So they're essentially inaccessible for studying in humans, right? There's no way to study that. And in fact, even just studying human interneuron or inhibitor neuron migration has been very, very difficult in humans, again, for very obvious reasons. So it's been, very interesting to not just discover these phenotypes, but then, because these models are, very tractable, they're scalable, you can now dissect the molecular mechanism. So we spend years now understanding precisely how that calcium channel causes that specific defect. And, it turns out that it's, through multiple pathways, and you have to modulate them. Experiments that would have been very difficult to do in any other way. And I think that's one example of a disease that we've been studying, and that certainly at this point, we've discovered strategies where we can restore it, many of the phenotypes, and so there is some promise for a potential therapeutic avenue at this point.
So, one of the things that really struck me in your talk that you gave here at Sunposium was, you showed images of cells migrating in the sort of the disease phenotype and the control phenotype, and it's very striking. But you also sort of emphasized that just by looking at this from a different perspective, using this system, like there's all these other surprising results, like just in terms of the mechanical aspect of how the cells are migrating. So making basic scientific discoveries just from taking a different perspective. Are there any examples of this?
Oh, yeah, totally. First of all, human cells are much, much larger. And the distances that they have to undergo in the nervous system to migrate, they're just at another level. So there are all kinds of adaptations that these neurons had to. So for instance, their processes are much, much longer in humans versus the size of the soma. The kinetic of that migration, so the cells like essentially jump. They don't crawl on a surface, but they pretty much jump every 30 minutes. In humans, it takes about 3 hours for them to kind of like decide in which direction to go. In mice, in 30 minutes, it's all done. Like they've undergone already the migration. So we're just talking about like very different scales. And what is interesting about like discovering like new phenomena is that of course we've been studying Timothy syndrome, which is caused by one mutation in one gene, but there are hundreds of genes associated with autism and neurodevelopmental disorders, literally hundreds. At least 100 plus high confidence autism genes, but probably close to 500, 600 genes associated with autism today. So one question that we've been trying to answer over the past few years was, could we actually identify, out of all those 400 plus 500 genes associated with autism and neurodevelopmental disease, which ones interfere with this process of interneuron migration and generation? Because interneurons and GABAergic cells have been historically associated with autism as part of the so-called excitation to inhibition imbalance in autism. So we took an unbiased approach, and we've done a CRISPR screen to identify all the genes that interfere with this process. And as part of this unbiased approach, we've discovered, for instance, that one very interesting gene called LunaPark, which is a endoplasmic reticulum structural gene. So, completely unexpected. You wouldn't expect that would interfere. And it turns out that as we were studying the role of this gene, that the ER, the endoplasmic reticulum, is actively involved in this interneuron migration, has a very interesting movement. And so I think this illustrates that, we're at the point where we can gain access to some of this quite complicated processes that underlie the assembly of the nervous system. Recapitulate them in vitro, certainly not in a complete way, but in a tractable way that allows you then to screen genes, identify the role of those genes in disease, and sometimes even discover, new aspects of human brain development, such as the involvement of DR in interneuron migration, for instance.
That's great.
Yeah, I think you've we're almost out of time, but I think you've really beautifully illustrated how taking this sort of unique approach allows us to understand both fundamental things about, neurodevelopment as well as potential for understanding neurodevelopmental disease. And another aspect that I love about your work is that you are really interested in educating others about using these approaches as well. And I believe you even have started a course to do that.
Yeah. So, I mean, one of the exciting things has been that, these technologies have triggered a lot of interest. I think people have a lot of questions to ask about human neurobiology and human neuroscience and, try to understand disease. And so we've been literally flooded over the past 8-9 years with requests for people to come to Stanford and learn the technologies that we've been developing. And we certainly help, over 200 labs, very often one-on-one. But it became clear at one point that we still, cannot do enough. So we started, as part of the program that I run at Stanford, we started an international course where we bring 20, 25 students at a time from all over the world for like 1 week of immersive organoid and assembloid, where we essentially, it's almost like a cooking show because, we have to prepare before, these experiments are hundreds of days long, but we show the critical steps and then we provide guidance, and of course we stay in touch with most of those students. So we've had, had students from all over, from Australia, from New Zealand, from India, from parts of Europe all the time, and creating a community. I mean, there's so many questions, right, to answer, and psychiatric disorders are, will take a very long time to fully understand. So I think this will be a community effort at the end of the day.
That's fantastic. Dr. Pasca, thank you so much for joining us on this podcast. It's really great to talk to you.
Thank you so much.
So we have these amazing speaker lineups here at Sunposium every time, and we're lucky as podcast hosts that we get to talk to some of these folks and really hear about the science behind their, what they're presenting. What were some of the cool takeaways about that you guys took from these interviews?
Well, from our first guest, Denise Cai, I think one of the really interesting things is how, the science drove the technology development. So she was interested in how memories are formed and how they're related in time, and then that spurred the development of their miniscope. And it's kind of cool to see how that has sort of gone beyond their science and affected other labs, and how Denise is also now using that to look at new questions about aging.
And we also got to talk to Dr. Ugur Dag, and he was actually one of the people who submitted an abstract and was actually selected by our selection committee to come and give a talk. And I think it's really exciting that we have an opportunity to highlight some of these really future up-and-coming scientists who are going to be the next leaders in the field. And his work was really incredible and interesting. I loved how he took this, tried to simplify everything in this very simple system with C. elegans, looking at the activity of just a single serotonin neuron and how it had really actually complex actions throughout the whole brain and whole nervous system to affect behavior.
So many of the talks this year were so mind-blowing. For me, several months ago, I heard this story on NPR about people using brain organoids to study neural development, and to have a chance to talk to Professor Pasca about his research was really, really interesting to me. Obviously, neural development is very complex, and there's all these different aspects of cell migration and synapse formation and refinement that we study in, lots of different model organisms and systems. But for him, wanting to study human brain development is an extreme challenge. So he takes this really sort of interesting, novel approach to do that in vitro in the dish, and he can answer new questions about all of those areas: how cells move from one brain area to another during development, how that's impacted by genetic mutations associated with neuropsychiatric disorders. Amazing work, and we thank all of our guests for sitting down and talking to us. This was a really great experience for us. Sunposium, we're sort of wrapping up here, and I think there's so many really interesting aspects of this conference that maybe aren't obvious on the surface. You think about the amazing speaker lineup, you think about the poster sessions, the stuff that you normally get at a conference, but I don't know, what are some of the things about Sunposium that you think really made this year particularly special?
Well, I particularly like the fact that we, every time we have Sunposium, highlight a young investigator through the Peter Gruss Young Investigator Award. And this year's winner, Dr. Michael Yartsev, I think was just incredible.
Yeah.
His talk was amazing. He studies this really naturalistic behavior of bats, and so he's actually looking at neural activity in free-flying bats, and he really made a big point about how behavioral preciseness is really key to understanding and studying neural activity robustly. And he challenged sort of some of the fundamental assumptions in the field about spatial navigation, and I thought that was just really fascinating and wonderful.
And I think even beyond the invited speakers, we also have a bunch of wonderful sponsors who help us run this event. And they also send representatives, so there's an opportunity to sit down with the folks that are helping you do the science, talk about like where they're headed with their development of technology. So had a nice chat with the guys at Thor Labs about what they're building now, and also it's a good opportunity to let them know about like where your needs might be in the future. And it's really a unique opportunity that you don't normally get.
Definitely. I mean, there's so many things about this conference that I really enjoy, like just being here and being a part of. One thing that, I find very personally satisfying is, working on training programs with MPFI now. We have a Florida Fellows program, so undergraduate institutions from around the state, we sponsor their students to come here, pay their registration, pay their hotels to stay in West Palm. We had 39 students come this year and 7 faculty, sort of chaperones for those programs. So this really opens the door to, I think, really cutting-edge neuroscience to students from across the state of Florida at schools where maybe they don't have quite the level of exposure to some of this research. I think, that's one of the things we're really trying to do is not just, put on a conference that's fun for us to have here in Florida and bring a lot of our esteemed colleagues to the area, but really try to elevate neuroscience in general in the area and create opportunities for students. So, I think that's one thing that MPFI can be really proud of for putting on a conference like that. And I mean, shout out to MPFI who employs me, of course, but
also all the other people that you don't see in front of the camera that are putting things together for this, right?
Oh my gosh. This was the first year I've seen Sunposium really come together on the organizational side, and just the amount of shout out to all the people involved. I'm not going to name names right now just because I don't want to leave anybody out. It's such a huge team effort. But to put on a conference of this size and scale and magnitude, there's so much going on behind the scenes at all times. Like there is, like the text chains and the coordination is just it's off the hook. It's amazing. I actually just said off the hook, so I think for the first time since like 1997. So a great conference. I had a lot of fun, and it sounds like you guys did too. So parting words of advice. We're going to do this again in 2025, right? I assume so.
We hope so.
Yeah, I hope so. So be on the lookout. 2025, usually around February, March, we'll have a really nice conference here in West Palm, and hopefully we'll see you there. Bye, everybody.
Thanks for watching today.
If you like this video podcast, make sure to subscribe to our YouTube channel for more cool neuroscience.